Okay, so these are the chapter 3 study points. Go through them really quickly. So what is cell theory, and describe the basic tenets. So basically, cell theory is that all cells come from other cells. That's the basis of biogenesis. And number 2, cells are the fundamental units of organisms. So Robert Hooke, we already discussed who he is and what he does, but basically, to reiterate, he introduced cell theory, and he was the first to visualize cells, fungal hyphae, and spores. And this was through the compound microscope, which he created. So when he looked at cells, the reason why it's called cork cells, or where that term comes from, is that when he looked at it, it looked like layers of cork to him. Understand the endosymbiotic theory. So endosymbiosis is where one organism is living inside the other. So think of it as like an organism engulfed the other one, and now that one's living inside of it. So basically, an endosymbiosis is where both organisms benefit, right? And basically, the theory is that eukaryotic organelles were formed by endosymbiosis. So the evidence is that bacteria share similar mitochondria characteristics with bacteria. So, or with eukaryotic cells. Sorry, I miswrote it. So basically, bacteria and eukarya kind of have similar mitochondria characteristics. They kind of look the same, so that's why they came up with endosymbiosis, or that theory. Describe how prokaryotic and eukaryotic cells are similar and different. So basically, this is something you really need to know for chapter 3. So prokaryotes, they're non-membrane bound. You need to know that. With singular circular chromosome. So basically, it has one circular chromosome, right? With 70 S ribosomes, and this is for bacteria and archaea. While eukaryotes, which is also known as like human cells, are membrane bound with 80 S ribosomes. And why is it important to understand the differences and similarities between the types? So for certain antibiotics, they target different structures of bacteria cells, right? So since prokaryotic cells have that 70S ribosome, basically what happens is that the antibiotics target that 70S ribosome instead of the 80S that are in eukaryotic human cells, right? So new antibiotics need to kill the bacteria cells, not the human cells. So that's why that is important to know. Know the structural features of prokaryotic cells and their functions. So they are flagella, pili, capsule, or cell wall. So flagella basically provides motility. Pili allows cells to adhere to surfaces. Capsule is basically, if it's an encapsulated bacteria, it has an outside layer that is slimy and provide bacteria to adhere to surfaces and provide protection. Cell wall is either thick or thin layer peptidoglycan. Describe prokaryotic cell shapes and arrangements. So the only ones that you really need to know is cocci and bacilli. Cocci, round and circular, bacilli, rod-shaped, and you can know the other ones, but cocci and bacilli are the most important ones to know. And they either come in chains or clusters. And what is meant by pleomorphism? Explain how this can affect your analysis of bacteria sample. So pleomorphism is basically like cell morphology. It's like the variation in cell shape and size. So it's important to know pleomorphism or be able to identify different cell shapes and sizes, because certain cell shapes and sizes are associated with certain species. So think of Staphylococcus, right? So Staphylococcus, think of the word. At the end of Staphylococcus is coccus, right? So it's going to be a round shaped bacteria. So it just aids in identifying bacteria under a microscope. Understand what happens to cells in hypertonic and hypotonic. So isotonics were no net movement. Hyper and hypo, I think of like two different scenarios. So hyper, I'm thinking like, ooh, I'm so hyper right now, I want to go to a party. So basically, it's kind of like H2O wants to go out of the house. So H2O moves out of the cell, which is a house, the house, right, to go party. And then what happens? The cell membrane shrinks and plasmolysis happens, right? Hypotonic, I think of like hippo, like round and big, right? So basically H2O moves into the cell, and it's like basically the cell kind of expands. However, it does, because of the cell wall in prokaryotic cells, it prevents swelling and lysis. So that's how I remember the two. Explain the advantages of cell walls. So basically, cell walls protect cells from changes in osmotic pressure and allow cells to maintain shape. So unlike human cells, where hypertonic and hypotonic would either like lead to cell death, the cell walls in prokaryotic cells actually protects the cells from dying completely. What is plasmolysis? So plasmolysis is shrinkage of a cell due to water being removed in a hypertonic solution. How does the concept of plasmolysis relate to food preservation? So plasmolysis actually aids in preservation. So when you think of high concentrations of salt, right, that acts as a preservative for bacon. And then high concentrations of sugar is preservatives for jams and honeys and etc. Understand the structure and function of the plasma membrane. So the plasma membrane is made up of phospholipids, which makes it selectively permeable. And it also has glycoproteins that act as basically cell towers for cell-to-cell communications, sensing and virulence factors. And also, it makes it identifiable. The cell wall of the plasma membrane of the prokaryotic cell makes the cell identifiable by the human body as well. Describe the molecular features of phospholipid molecules. So plasma membrane, like I said, has a phospholipid bilayer that makes the wall semi-permeable. And that's made out of a hydrophobic tail, which is going to be the inside, pointing towards the inside of the membrane, and then a hydrophilic head, which is towards the outside of the cell membrane. And what does it mean for the plasma membrane to be selectively permeable? So basically that means that it prevents molecules bigger than water. You need to remember that, and that are like not charged to enter through simple diffusion. What kind of molecules can or cannot diffuse across the cell membrane? So basically, what can enter are gases, water, small molecules, and molecules that aren't charged. What cannot enter are any molecules, like I said earlier, that are bigger than water. So sugar molecules, sodium, potassium, any charged molecules. And simple diffusion is basically you have a cell that, or molecule that has no energy, smaller than water, can go through the membrane without any assistance. Facilitated is where it is like a charged molecule, and for larger molecules, but only require transport protein. And then for active transport is where it needs a transport protein and energy, aka ATP, to move against concentration gradient into the cell wall. Peptidoglycan, structure, function, synthesis, unique features. So peptidoglycan is only in bacteria. You need to know this. Composed of 2 glycan chains of NAM and NAG sugar across a tetrapeptide chain and a peptide in a bridge. And only a peptide in a bridge in only gram-positive bacteria. And you do need to know the structure of the NAM and NAG sugar on the test. It does show up. And chemicals that affect peptidoglycan structure. So certain antibiotics like penicillin target peptidoglycan synthesis. And why is that effective against bacteria, not human cells? Is because why? Peptidoglycan is only in bacteria. Chemicals like lysozyme also destroy the integrity of the glycan chain, which is only in bacteria, not in human cells. So gram-positive and gram-negative cells will understand the structure, similarities, and differences. So gram-positive is a thick outer wall of peptidoglycan. It has teichoic acid, structurally simple, more porous, and fully permeable. That's why it's easier for the gram stain to retain in gram-positive bacteria. For gram-negative bacteria, it has a thin layer peptidoglycan, and this is really essential to know. It has the outer layer of lipopolysaccharide. So that makes it more complex, and it acts as a barrier to some antibiotics. That's why gram-negative is harder to kill than gram-positive bacteria. Describe the structure of the gram-negative outer membrane. So, like I said, it has lipopolysaccharide, which has a lipid A and O antigen. LPS, basically, lipopolysaccharide, it has a lipid A portion that acts as an endotoxin and causes like vomiting, throwing up, all the symptoms, and an O antigen. What is a glycoalix? You need to know this one as well. Explain the different types and their features. So a glycoalix is basically like a jelly-like substance that is secreted out of a cell that allows capsule to attach to the cell wall. And this is only in some bacteria, right? So it basically is involved in biofilm formation and kind of acts as an invisibility cloak that prevents the encapsulated bacteria to be identified by the body. So basically, it's really important to know this because it makes it harder for antibiotics to target this bacteria and for it to even be detectable by the human body. Because since it's that slimy layer, it makes it harder for the antibiotics to attach to the bacteria, if that makes sense. What is a bacterial biofilm? So this is a microbial community that provide protection and keeps the bacteria from drying out, associated with chronic infections, prevents antibiotic penetration, and hard to get rid of. So think of like dental plaque. And that's from that biofilm that I talked about earlier. And what are flagella and pili and their function? I already went over this. Basically, pili is for attaching to surfaces, flagella is for cell motility. And then internal structures of prokaryotic cells. So basically, it has the ones that you need to know that are different from eukaryotic is that sometimes it has plasmids, which are extra chromosomal DNA, ribosomes, that 70S ribosome, and endospores, and which is basically an armor against extreme environments, which I'll get into later. And what is the structure of the chromosome in bacteria? So the structure of bacteria is that it's double-stranded, and like I said, it's a single circular molecule of DNA, while as eukaryotes are multiple. So instead of the single circular single molecule of DNA, the eukaryotes have multiple. That's like the biggest one you can remember to differentiate the two. And what is a nucleoid? So the nucleoid is basically the location of all or most genetic material, lacks a membrane, membrane bound in prokaryotic cells. While, what's it called? Like I said before, eukaryotic has that membrane bound nucleus. Plasmids, what are they? So plasmids are basically extra chromosomal DNA that's not essential but can aid in cell survival. So basically, it's not needed. You need to know that. It's not needed, but it can aid and code for resistance to antibiotics, disinfectants, or able to produce certain toxins. And explain how prokaryotic and eukaryotic ribosomes differ. How are they similar? Like I said, prokaryotic 70S ribosome, and they also have that 16S rRNA. Eukaryotic, that 80S ribosome, and have that 18S rRNA. What are the differences between prokaryotic structures? So basically, you need to know why, essential to know like how an antibiotic should target, certain bacteria cells, right? Because of that 70S ribosome. Storage granules, inclusions, you don't really need to know this that much, but basically it's just where extra nutrients are stored in like the cell. Endospores are, you need to know this. Endospores are basically a thick, dry wall of peptidoglycan, so like a sheet of body armor around the cell, that basically makes the bacteria more resistant to heat and harsh chemicals, which basically allows it to live in extreme environments. So, for example, if a bacteria is in a place where it's super hot, it's super in a place where there's a lot of harsh chemicals, it can remain dormant, right? So it's not gonna be active, but once the environment is like less hot, it's nicer, then that's when the bacteria would come out of hibernation and be active. That's basically what it means. It basically protects the bacteria from these super harsh extreme environments until it's ready to come out in a nice environment where it can thrive and grow. Now, know the genus, species, the endospore formers we discussed in class. You need to know these ones because these are important diseases that are hard to get rid of in a hospital setting. So like anthrax, hard to get rid of, gastric gangrene, botulism, tetanus, C. diff especially. You need to know these because they are hard to kill because of that endospore that is in that bacteria. And then understand the case study. So this case study, basically, there's a patient with pneumonia and they were given the wrong antibiotic initially. The antibiotic was amoxicillin, which is basically a cell wall synthesis inhibitor. Why didn't it work? It did not work because pneumonia is a mycoplasma that lacks a cell wall. So why amoxicillin will not work against bacteria that doesn't have a cell wall, because there's no cell wall to inhibit. Does that make sense? So eventually what happened was that they gave azithromycin, which targets like bacteria ribosomes, the 70S ribosomes, and eventually cured the patient. So yeah, that is it.